Hi everyone. In this first lecture of this course, we will see what is transcription and introduction to transcription. So, what you see on the screen is the central dogma. 1956, 3 years after Watson and Crick proposed their double helical structure of DNA, Francis Crick proposed this two-step process called a central dogma. The central dogma is beyond this two-step process, but we will focus on these two steps. The first step is the information in the DNA, that is the sequence information of the DNA, is used to synthesize messenger RNA. This process is called as transcription, and this entire course is all about this step. In the second step of the central dogma, the information in the mRNA is converted to the sequence information of the protein. So we all know that double-stranded DNA is the genetic material in most of the organisms on the planet. And in transcription, one of the strands of the DNA is used as a template to synthesize messenger RNA, and the sequence of the gene, the sequence of the DNA decides the sequence of the mRNA. And then in the second step of the central dogma, translation, the sequence of the mRNA decides the amino acid sequence of the protein. This is also called as gene expression. Transcription is the first step of gene expression, and translation is the second step of gene expression. So when I say that a gene is being expressed, which means the gene is being transcribed. So this transcription is the first step of gene expression, and this translation is the second step of gene expression. Let's see more about transcription. This process of transcription, that is synthesis of messenger RNA from DNA, can it be done without the help of an enzyme? No, it's an enzymatic process. We know enzymes are biocatalysts. Enzymes are present in all living organisms. And this is the enzyme that you can read here is involved in the transcription, that is the synthesis of messenger RNA. So let's dissect this name of this enzyme. What does it do? So the last word here is polymerase. The last three letters of this word, ASE, makes us understand that it's an enzyme. Polymerase means it's an enzyme that synthesizes a polymer. Which polymers? If we see that the end result of transcription is synthesis of mRNA, which means a type of RNA is being synthesized. So this polymerase is naturally synthesizing RNA. But there are two more words before the RNA polymerase, which is the transcription enzyme, which are DNA dependent. What do you mean by DNA dependent? DNA dependent means that this enzyme, polymerase, synthesizes RNA. So it's an RNA polymerase, but it synthesizes RNA using DNA as a template. When the transcription occurs, RNA is synthesized from DNA. So since DNA is used as a template, the enzyme is called as DNA-dependent RNA polymerase. In this nomenclature, when you will learn some other enzymes as well, you will see that the second word is always dependent. The last word is polymerase. And the third word, which is the word just before the polymerase, is the molecule which is being synthesized by the enzyme. And the first word is the molecule which is used as a template. So in this picture, there is a DNA molecule, and a part of the DNA is shown green in color, which we'll see in the future lectures. and the rest of the DNA is shown in pink color. And on this DNA, a big yellow color thing is sitting. This is the DNA-dependent RNA polymerase enzyme, which is doing the transcription and which we just spoke about. The DNA is denatured. Let's see the structure of the DNA. The DNA is double-stranded. It's helical. One strand is wound over another. The strands are anti-parallel. So one strand runs in 5' to 3' direction, and the other strand runs in 3' to 5' direction. Inside, there are nitrogen bases. There are 4 nitrogen bases in DNA: adenine, thymine, guanine, and cytosine. Adenine pairs with thymine with 2 hydrogen bonds. Guanine pairs with cytosine with 3 hydrogen bonds. And outside is the sugar phosphate backbone with phosphodiester bonds. During the process of transcription, when the DNA sequence has to be read and the sequence dictates or decides the sequence of the mRNA, DNA has to be denatured. You can see the denaturation, and by denaturation, the 2 strands separate. A local transcription bubble is formed. This region is denatured. And when the 2 strands are separated, they unwind over each other. The hydrogen bonds between the nitrogen bases break and the strands are separated. And this denaturation or the separation is brought about by none other than this DNA-dependent RNA polymerase enzyme. And this light green color thing is the mRNA which is being synthesized. And this mRNA is bound to this one of the DNA strands. So it's called as RNA-DNA hybrid. Now this mRNA always has the polarity 5' to 3'. So the mRNA always has the polarity 5' to 3', which means that the transcription takes place in the 5' to 3' direction, and the mRNA has the polarity 5 to 3. Since mRNA is synthesized in 5 to 3 direction and has the polarity 5' to 3', the DNA strand from which this mRNA is made, the DNA strand has the polarity 3' to 5', and this is called as the template strand, the strand that is used to synthesize the messenger RNA. And as we all know, only one of the strands is used as a template for mRNA synthesis, not both the DNA strands, for a gene to be transcribed. And the opposite DNA strand, which has the polarity 5 to 3', which is complementary to the template DNA strand and has the same polarity as mRNA, this 5 to 3 DNA strand is called as a non-template strand. You can see here it's mentioned, this one is template strand, this one is non-template strand. And in the next slide, there's a lucid drawing of the same thing, that mRNA is being synthesized in 5 to 3 direction and has the polarity 5' to 3'. And the DNA strand, which is used to synthesize this mRNA, has the polarity 3 to 5 and is a template DNA strand. And the other DNA strand has the polarity 5 to 3. It's complementary to the template DNA strand, has the same polarity as the mRNA, and it's called as a non-template strand. The RNA precursors for transcription are the ribonucleoside triphosphates, ATP, GTP, CTP, and UTP. The precursors to synthesize RNA are the ribonucleoside triphosphates, collectively called as nucleoside triphosphates, and adenine, guanine, cytosine are there. But as we all know that there is no thymine in the RNA and there is uracil instead. RNA synthesis occurs by polymerization reactions similar to those involved in DNA synthesis. RNA polymerase reads the sequence on the template DNA strand, selects the next nucleotide from the pool to be added to the chain by its ability to pair with the exposed base on the DNA template strand. As we already said that RNA chain contains nucleotides with the base uracil instead of thymine, and uracil pairs with adenine. So wherever there is adenine on the DNA template strand, instead of thymine, a uracil is placed in the mRNA. So let's see an example. if the template DNA strand reads this sequence, 3' to 5', ATACT, double GAC, the RNA chain will be synthesized in the 5 to 3 direction, will have the polarity 5' to 3', and will have this sequence. So the first nucleotide in the DNA template is adenine. So there will be no thymine, mRNA will have uracil. Then opposite thymine, adenine will be added. Again, there is an adenine in the template DNA, so uracil. The cytosine in the template DNA, so guanine. There's thymine in the template, so adenine. The two guanines, so two cytosines in the mRNA. Second last nucleotide is also adenine. So there is uracil added in the mRNA. And then guanine, since the template DNA has cytosine. So in this first introductory lecture, we have learned what is central dogma, what is transcription, how is it important, the enzyme involved in transcription that synthesizes the messenger RNA, DNA-dependent RNA polymerase, in which direction mRNA is synthesized, what is template strand, what is non-template DNA strand, and how thymine is replaced by uracil and the polarity.